I'm Trey and this is Cyrus, and we're going to talk to you about DNA and the big three. Cyrus is going to start you off with the first step in it, which is replication.
Before DNA can be replicated, the double-stranded molecule must be unzipped into two single strands. DNA has four bases called adenine, thymine, cytosine, and guanine that form pairs between the two strands. Adenine only pairs with thymine, and cytosine only binds with guanine. In order to unwind DNA, these interactions between base pairs must be broken. This is performed by an enzyme known as DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into a Y shape known as the replication fork. This area will be the template for replication to begin.
DNA is directional in both strands, signified by a 5' to 3' end. This notation signifies which side group is attached to the DNA backbone. The 5' has a phosphate P group attached, while the 3' end has a hydroxyl OH group attached. This directionally is important for replication as it only progresses in the 5' to 3' direction. However, the replication fork is a bidirectional. One strand is oriented in the 3' direction, 3' to 5' direction. The leading strand. Well, the other is oriented 5' to 3', which is the lagging strand. The two sides are therefore replicated with two different processes to accommodate the directional difference.
The leading strand is the simplest to replicate. Once the DNA strands have been separated, a short piece of RNA called a primer binds to the 3' end of the strand. The primer always binds as the starting point for replication. Primers are generated by the enzyme DNA primer.
Enzymes known as DNA polymerase are responsible for creating the new strand by a process called elongation. There are 5 different types known as DNA polymerase in bacteria and human cells. In bacteria such as E. coli polymerase 3, and the main replication enzyme, which is polymerase 1, 2, 4, and 5, are responsible for air checking and repair. DNA polymerase number 3 binds to the strand at the site of the primer and begins adding new base pairs complementary to the strand during replication in eukaryotic cells. Polymerase alpha, delta, and epsilon are the primary polymerases involved in DNA replication. Because replication proceeds in the 5' to 3' direction on the leading strand, the newly formed strand is continuous.
The lagging strand begins replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerase then adds pieces of DNA called Okazaki fragments to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjointed. Once both the continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strands. These primers are then replaced with appropriate bases. Another exonuclease proofreads the newly ligase joins Okazaki fragments together, forming a single unified strand. The ends of the linear DNA present a problem as DNA polymerase can only add nucleotides in the 5' to 3' direction. The ends of the parent strand consist of repeated DNA sequences called telomeres. Telomeres act as protective caps at the end of chromosomes to prevent nearby chromosomes from fusing. A special type of DNA polymerase enzyme called telomerase catalyzes the synthesis of telomere sequences at the ends of the DNA. Once completed, the parent strand and its complementary DNA strand coils into a familiar double helix shape. In the end, replication produces 2 DNA molecules, each with one strand to the parent molecule and one new strand.
So basically, just to sum up what replication is, DNA replication is the production of identical DNA helicases from a single double-stranded DNA molecule. Each molecule consists of a strand from the original molecule and newly formed strand. Prior to a replication, the DNA uncoils and strands separate. A replication fork is formed, which serves as a template for replication. Primers bond to the DNA in DNA polymerase, add new nucleotide sequences in the 5' to 3' direction. The addition is continuous in the leading strand and fragmented in the lagging strand. Once elongation of the DNA strand is complete, the strands are checked for errors, repairs are made, and telomere sequences are added to the ends of DNA.
The next part of the big three is transcription. The first step to transcription is initiation.
Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. The ribosome binds to the mRNA at a specific area. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. The next step in translation is elongation.
Elongation is the addition of nucleotides to the mRNA strand. RNA polymerase reads the unwound DNA strand and builds the mRNA molecule, using complementary base pairs. There is a brief time during this process when the newly formed RNA is bound to the unwound DNA. During this process, an adenine in the DNA binds to a neutral in the RNA. The next step is the transcription is termination.
Termination is the ending of transcription and occurs when RNA polymerase crosses a stop sequence in the gene. The mRNA strand is complete and it detaches from DNA. The last step of the big three is translation.
First, the ribosome binds to mRNA as a specific area.
The ribosome starts matching tRNA anticodon sequences to the mRNA codon sequence.
Each time a new tRNA comes into the ribosome, the amino acid that it was carrying gets added to the elongating polypeptide chain.
The ribosome continues until it hits a stop sequence, then it releases the polypeptide and the mRNA.
The polypeptide then forms into its native shape and starts acting as a functional protein in the cell.
And this is an animated video to hopefully help you process the big three. Thank you for watching. I hope this helped.